27 or 22.
Yeah. Welcome. Welcome to the Scientist Podcast. And I'm joined by my co-host, Mrityunjay Sharma, obviously. And today we have a special guest, my friend, I will say from childhood because I'm very old. So when I was in 11, 12 in DAV, Shamli, JVM, just those who don't know why this school is famous, because MS Dhoni was there. And he
And
also
he also came to our class once while
and
we were studying.
she was front bencher, I was last bencher, so she could shake him, I could not. So because we had an ego, yeah, kaun jata hai, something like that. But after that, she has done an amazing job in research. She has like almost 13 years of experience in research, mostly in life sciences research. She has been to TIFR and all biggest institutes of India. Then she moved to Germany, and she is there since last 10 years. And yeah,
10 years now. Yeah.
in Hanover Medical School, one of the best medical school in the world, not only in Germany. Everyone knows that. My PI from here, he knows it, everything about it. And then she's mostly focusing her work on telomerase and also in heart failure. And her lab works on the drug development for different kind of cardiovascular diseases, which is one of the highest causes of mortality across the world and very understudied study in India. I don't know why every city is not studying that. It's very unique because India is top in that pile. She has almost 3 dozens of paper, which is a lot for a young scientist. Like a lot. That means she doesn't sleep. First, second, and those papers are verified by almost 1,500 citations. So today is gonna be a learning experience for all of us. So thank you, Shambhavi, for joining us. And please just tell me about yourself from your childhood to how did you come up here?
First of all, thank you for this very elaborative introduction for me. I hope I can do justice to all the good words you said. Um, my journey, I think, in science, if I really remember, like the first memory when I got interested or fascinated with biology was when I was about 3 or 4 years old. And my mom, my grandma, like all the women in my family love gardening. So I think she was repotting a plant, and that was the first time I saw, like, okay, underneath the green stuff, there are roots. And as a 3 year old or a 4 year old kid, that like blew my mind. I was like, wow, this is so cool. I think that was my first interest into biology, like how things grow. And then around 4, 5, 6 years old in school, I think for kids in our generation, they will remember CID, Dr. Salunke.
Yeah.
And I think that was my first memory of seeing like a microscope on television and hearing the words like DNA and blood sample. That sort of like interested me a lot, like how you can bring justice to people with a microscope in hand. And over the years, like in school, the more I learned in science, I always like found it to be very interesting. But I soon realized botany is not for me because you have to memorize a lot of names. And I was like, no, this is not going to be fun. I don't want to keep memorizing 10,000 Latin and Greek names all my life. So that was like a clear no for me. But I still enjoyed learning about how things grow and regenerate and develop. So that was something I knew I wanted to do. And growing up in my city, like, I mean, I was born in Patna, then I moved to Ranchi when my father moved there. And in that era, I would say the only options you had was if you are interested in biology, you become a doctor. Like, that was it. There was no other, let's say, idea of what else can be done in science. Um, Having known only the option of being a doctor, I was like, okay, I will study biology in 12th grade and then go for medical exams. While I was doing that, I didn't qualify the first year into the schools that I would have liked to go to. And then I was like, okay, what else can I do? And at that time, there was in St. Xavier's College, Ranchi, the biotechnology department had just started. So I was the second batch of student in the St. Xavier's College, Ranchi biotechnology department. And then when I started studying there, I was like, oh, this is actually quite nice. And then you can do research. That was for me the first time. I was like, oh, there is like a lot of research that happens, which you can then use to make drugs. And then these drugs go into the clinics where patients are treated. So for me, that was like, this would be like a back way for me to help patients at the end.
Hmm.
If I cannot be a doctor and do it in person, this is how I can help and contribute to the society. So that like sort of nailed it for me, like, okay, if I cannot be a doctor, then this is what I want to do and this is how I want to help the healthcare system. And after that, I think it was more of a journey for me to figure out what in research I would like to do. So did my master's, then I studied microbiology and genetic engineering. Then I worked for 2 years in TFR in Professor Krishanu Ray's lab. I mean, now he retired last year, but I think those 2 years in Madurai Kamraj University during my master's, and then the 2 years that I worked in TFR were like the foundation for me as far as being a researcher is concerned. And then all the learnings I had there from both these places helped me a lot during my PhD.
Mhm.
And then during my PhD, I again then was like figuring out what I want to do. So as I said in masters, it was genetic engineering and microbiology. I had a feeling like it was not so application based. Like, my aim was to help the patients at the end.
Mhm.
Then I studied, when I was in TFR, I was doing a lot of developmental biology and cell biology. I enjoyed the project a lot. I was really happy in the project that I was doing. But again, I had this feeling like, this is not going to help the patient directly at the end. And
Mhm.
so I was looking for options where I could like work in projects that are more application based. And
Mhm.
then I got to apply for a lot of different PhD programs, and one of them was Hanover Medical School, which I was lucky enough to get selected through the interview rounds.
Mhm.
And here, the project I was getting was like the most interesting for me because I felt this would bring me the closest to having a direct effect in the clinics.
Mhm.
This is how I ended up choosing Hanover Medical
Mhm.
School for my PhD. Did my PhD here, then I
Mhm.
defended in January 2020, and then COVID came.
Mhm.
And then whatever I had planned for my postdoc sort of had to be rescheduled.
Mhm.
Plan A didn't exist anymore, had to go with plan B, sort of stayed back in the same lab and then developed my own projects here.
Mhm.
So, yeah, this has been my journey in Germany for 10 years now that I've been in the same place.
Mhm.
And in that way, I've been lucky enough because I've been able to develop my projects further because I
Mhm.
stayed in the same place. So it was a lot easier in that perspective for sure.
And what was your project about in your PhD? And what is it like? What is it that you have been developing over the years?
So when I joined the lab, this is also funny because the during the interviews when I got the project proposals from different labs,
Mhm.
the project that I liked most was to study how telomerase can be helpful in cardiac regeneration.
Mhm.
And this is super important because our heart, which
Mhm.
is composed of many different cell types, the main
Mhm.
is the cardiomyocyte, which allows the heart to contract. So if the
Mhm.
cardiomyocytes are not there, the heart cannot pump blood day in and day out.
Mhm.
And these cardiomyocytes, if they die, they don't regenerate. So the human cardiomyocytes don't have regenerative potential. And this is why heart failure is such a big problem because once the cardiomyocytes are dead, there's no way to replenish them.
Oh.
Fibroblast, you can do it. Endothelial cells, to a certain extent, you can have angiogenesis. But for cardiomyocytes, that doesn't work.
Mhm.
And this was like for me, like, oh, super cool. I will be working on a way how you can make the cardiomyocytes proliferate again. And
Mhm.
telomerase was like a protein that I had already studied during my bachelor's. So
Mhm.
it is a very standard, if you have read biochemistry or cellular biology, you
Mhm.
have read about telomerase and telomere. So Professor Elizabeth Blackburn had discovered telomere as structures in tetrahymena long, long ago. She was even awarded the Nobel Prize for it. And these are basically protective ends of a chromosome. So all of us have chromosomes, which has
Mhm.
the DNA in it. And these DNA that encode for proteins, they are basically important for all the cellular function.
Mhm.
And every time the human cell divides, you would have the problem that some part of the DNA will be lost. And to protect the main or the important DNA information, the cell did a very clever thing. It had an enzyme called telomerase,
Mhm.
and this telomerase added extra base pairs to the ends of the DNA. So even though the cell divides, it will only lose the telomere, and the most important genes and the DNA will stay protected. And this is how, let's say, the cells are able to divide throughout our lifespan. When you have short telomeres, that means there is no telomerase activity anymore, so the protein is not there. And therefore, every time the cell is dividing, you have this shortening of the chromosome.
Mhm.
And at some point, the telomeres get so short that the cell either cannot divide anymore or the cell tries to reactivate telomerase, and then you end up with cancer because there you have high telomerase expression and the cell becomes more like a tumor and it constantly keeps proliferating. So telomerase is a protein that you only have during embryogenesis, so when the embryo is growing, so that it can add these telomeres to the chromosome. Once you are born, it stops doing its function, and then whatever you have, the telomere length reserve, is what you have for the rest of your life. And
Oh,
this is why you
wow.
have aging. Yeah.
And oh my God. So does that mean that so does that mean that these telomerase they are like can I actually look at the length of a telomerase or in a certain person and can I define how long they are going to live or not live?
Um, Telomere length is a predictor of aging and
Mm-hmm.
lifespan,
Mm-hmm.
100%.
Mm-hmm.
But just measuring the telomere length does not give you how long they will live. You also have to know how fast the telomere is getting shortened. And
Okay.
this then depends on your lifestyle. So if you have a healthy lifestyle, if you're not smoking, not drinking,
Mm-hmm.
eating well,
Mm-hmm.
then on average,
Yeah.
for a healthy human, you
Mm-hmm.
lose about 11 kb
Mm-hmm.
telomere length per year. That is
Mm-hmm.
the average, assuming you have a healthy lifespan, a
Mm-hmm.
healthy lifestyle.
Mm-hmm.
If not, it could be much more faster for you.
I have a very naive question, but I'm going to ask, right?
No, go
Like
ahead.
what is it like why does a bad lifestyle, why is the bad lifestyle causing the telomere to break down or to get shortened in length?
Yeah. So let's assume if you have unhealthy diet, or
Okay.
let's say if you live in a city which is very polluted, like there's high levels of air pollution. So
Okay.
the air you breathe
We
or the
know a few
food,
of those cities.
I know we all do.
Unhealthy,
Unfortunately for us, we all do.
unhealthy
Yeah.
diet, living in Delhi. Okay.
Yeah.
Either of the situations, what happens is your body is going to react to the pollutants or to the unhealthy diet, and you will have oxidative radicals in your body. So, superoxide radicals, which
Mm-hmm.
basically induce DNA damage. So, the
Mm-hmm.
DNA is made of four main bases
Mm-hmm.
ATGC, so adenine, thymidine, guanine, and cytosine.
Mm-hmm.
And out of this, the the G and the C, so the guanosine and the cytosine, are the most susceptible to any kind of oxidative stress.
Okay.
And the telomere sequence is TTAGG. So that means it has
Mm-hmm.
high number of G and Cs.
Wow.
The
Mm-hmm.
more stress you have, which could also be physiological chronic stress from your work, from your life, that can also lead to this oxidative damage in all the cells. It could be air pollution, it could be your lifestyle.
Yeah.
All of this, no matter from where it starts, it
Mm-hmm.
ends up into oxidative radicals in your cell. The
Yeah.
moment you have oxidative radicals, it will cause DNA damage,
Yeah.
and your telomeres, which are very G and C rich, will
Yeah.
have more of the DNA damage. So, this is how, even though you might have a long telomere length
Yeah.
at birth,
Mm-hmm.
if you have had a very unhealthy lifestyle or a very stressful lifestyle,
Yeah.
you had more oxidative stress,
Mm-hmm.
therefore, your telomeres shortened faster than the other
Yeah.
person.
Oh my.
So, it doesn't, this is why you can have a person of 50 years old who looks
Yeah.
very young, Milind
Yeah.
Soman, for example.
Yeah.
I don't know his exact age, but just giving an example here, or Cristiano Ronaldo, he
Yeah.
is very old but looks very young.
Yeah. Very healthy.
Otherwise, you could also have people, young people who are like 25, 30 years old, not giving out our ages. Abhilash, don't try that.
Oh yeah.
You
Oh
could
yeah.
have, on
If
the other hand,
your age,
people
shit.
who are quite young but still
But
look much older. They have gray hairs or they have very low in energy.
Oh
So
my.
all of this then is like a, yeah,
Uh,
indirect effect of telomere shortening in one. So it's
oh.
one of the ways that you have aging. It's
Wow.
not the only way, but it's one
Yeah.
of the most important ways your body is aging.
Wow, this is crazy because I am assuming like I think a lot of the times, right? This has been explained that hey, unhealthy lifestyle, you will have like, the mortality rate or whatever increases this and that. But the basic of it is this actually, right? And it is so simple, right? Just make sure that you're not having unhealthy lifestyle, and unfortunately we all do, right? In some form or way what we are doing, we are just causing damage to our body, right? Interesting.
I'll try to
Interesting.
do this after this podcast. So
Yeah, yeah.
so
So
that
I have another question though. How is it? How are these telomerase like what is their importance in cardiovascular disease? And people who are listening to us, right? Uh, please also define like what is a cardiovascular disease and like why are people actually, going through them and what is happening in that particular disease?
You have given me a very difficult task.
Oh,
Defining cardiovascular disease
okay.
is tough
so PA,
Okay.
because
Yeah.
because I guess Abhilash will hopefully agree with me. There are
Mhm.
many types of cardiovascular disease, so I cannot give
Mhm.
one
Fair point.
definition that tells
Fair.
like, oh, this is cardiovascular disease.
Hmm.
If I wanted to simplify it for the
Yeah.
audience,
Yeah.
any situation that does not allow your heart to pump blood properly,
Ah.
disease. Now, why is the heart not able to pump the blood? It
Mhm.
could be that the heart is super stiff as a muscle and it cannot pump blood. So, this can
Mhm.
happen in hypertrophy or cardiac fibrosis, which are types of cardiovascular disease.
Mhm.
You could also have heart failure with preserved ejection fraction. So, this is a very technical term, but that means that the heart is able to pump blood, but it still has other symptoms of heart failure. For
Mhm.
example, the blood vessels are blocked, or there's like
Mhm.
initial stages of heart failure where it
Mhm.
is still a bit reversible. So, if I
Mhm.
had to easily define it, any
Yeah.
situation where your heart is not pumping blood as it should, it's a
Yeah.
heart failure. For example, there is also atrial fibrillation, which is a cardiovascular disease where
Mhm.
your heart is pumping blood, but there are moments where your heart is beating super fast
Mhm.
or
Mhm.
beating arrhythmically. So, it's not beating in a normal rhythm.
Mhm.
Again, a cardiovascular disease. So,
Interesting.
very
I have
hard to
a
define it in one line.
Yeah, yeah, sure, sure. And I just wanted to, before we go on further, just wanted to clear one more thing. So these telomerase, right? They are also non-reversible, right? Once they get divided, they get divided, right?
Yeah.
You also used one more word when there was one more thing, cardios, some like I don't remember what you were talking. So when you were talking about when the when the heart pumps blood, there is some cell that is also irreversible, right? If it
The
leaves,
cardiomyocyte, yeah.
yes,
So
the cardio,
the cells that
yes.
are making the
So
contractile
that's
force, yeah.
also reversible, right? You can't change that. So my question is like, this is very dangerous. I
I
mean,
know.
and why is no one telling me that this is irreversible, right? Like
oh,
this
no
is
one is telling.
crazy. I mean, everyone is like, do this, do that, but no one is telling that, hey, this is irreversible. Because being irreversible, right, I think it is the biggest challenge if I'm not being too naive, about this topic, right?
So everyone around the world is studying cardiovascular disease just because of that. Because once you
Yeah,
get it,
yeah.
it's done. So
But is there a
Someone
way
can
to?
design a way to rejuvenate the cardiomyocytes or to make them proliferate, Nobel Prize.
Oh,
100%.
yeah,
oh,
That's a $1 million question in the cardiac field.
yeah.
uh-huh.
How
That,
you can do that.
yeah, oh, interesting. And no one is able to do that
What
till now.
most people do, people have like in vitro cell cultures where they
Mhm.
convert normal cells into cardiomyocytes. They just learn their functions. That's the most they can do right now.
Oh,
So this is
wow.
a big part of our research in the lab, also cardiac regeneration. So what
Mhm.
I started with was the telomerase, having this in the idea that, okay, I know that the telomerase protein doesn't exist anymore. So
Mhm.
what if I give these cardiomyocytes, so the ones that don't divide anymore, if I give these cells the telomerase again,
Mhm.
will they start to proliferate? That was the one of the research questions I was trying to answer.
Mhm.
And this was actually, we had started this discussion because this was the project I was given in the proposal. So
Mhm.
my experiments would have been in animals, so in mouse model, to do
Mhm.
this exact experiment. You
Mhm.
have an adult mice
Mhm.
where you know that the cardiomyocytes, so the ones that help the heart to contract, they don't proliferate anymore, and you give them the AAV or telomerase reactivation using some viral gene therapy method, and then you observe if the cardiomyocytes can proliferate or not. In Germany, you also, if you work here in science, you have to follow a lot of legal regulations.
Fair
And
point.
to do any kind of animal experiments, you need to have animal licenses approved.
Mhm.
The time when I was going to start my PhD and with the postdoc at that time, and now he's the group leader and professor in our group.
Hmm.
He was also coming to join the lab. So we did not have all the approvals that would be needed to start with this experiment.
Hmm.
Therefore, even though I had said yes to doing this project specifically, I never got to do it in my PhD.
Oh.
Instead, what happened was I got shifted into the whole world of stem cells, which exactly what Abhilash mentioned now, that if you have stem cells, you can differentiate them into cardiomyocytes and then study what happens. So all my experiments then eventually went into more stem cells and stem cell derived cardiomyocytes.
Hmm.
And there we did these experiments to see if you give back telomerase to the cardiomyocytes, do they proliferate or not?
Hmm.
And for us, it was interesting to see that, yes, they do proliferate, but
Yes.
it's still only 1 or 2% more,
Ah, okay.
which
Okay.
is not helpful enough if you want
Hmm.
to help a patient who has
Yeah.
lost half of their cardiomyocytes in the tissue.
Yeah. Yeah.
So, as I said, this is still $1 million question. What
Hmm.
we do know is that when you're doing this differentiation, so when you're forcing a stem cell to
Hmm.
become a cardiomyocyte,
Hmm.
so for those who don't know what stem cell is, these are the cells that can divide into any cell type of the body. So they
Hmm.
would have the ability to become an eye cell,
Hmm.
they would have the ability to become a skin cell, a
Hmm.
heart cell, a kidney cell, a liver cell, whichever cell type you want to make, you should be able to differentiate it from a stem cell because these are the ones that are in the embryo also during
Hmm.
development.
Hmm.
And what we observed that if you did this telomerase reactivation earlier during the differentiation, so when the cardiomyocytes are still naive and not adult enough, then you can make them proliferate much more.
So in younger
That
mouse models.
sorry,
So in like younger mouse, not in adults.
this is all human stem cell in the cell culture.
Oh, okay. Okay. Oh, so this is happening in the cell culture. Okay. Interesting. Hmm.
And after my PhD was done, finally, by the time we got the approval for the animal experiment, there another colleague of mine, Alessia, she also did her PhD with in our group, and now she's a postdoc here. So she is doing those experiments, which were supposed to be my PhD experiment,
Hmm.
where we do the experiments now with the mice model. And there we clearly see that if you reactivate the telomerase to a certain time after birth, so the
Hmm.
first 5, 7 days, it works because there the heart is completely recovered, no problem at all. But if you do the same telomerase reactivation, so if you give back the telomerase protein after 7 days of the mice being born,
Hmm.
it doesn't, it helps, but
Hmm.
it's not like back to normal. So we know that there is a window at which the cardiomyocytes are still, uh, like you can still persuade them to proliferate.
Hmm.
Once they cross this time point, it's very hard to make them proliferate again, whether you have telomerase or not.
How strong is the this inverse correlation though? Like, so I'm assuming when they are really young, it's very easier, like it's easy enough, let's put it that way. But what is the threshold? Like, how does it drop down? Is it like very drastic, or is it like still like a easy curve going down?
I would say there's almost like a 50-60% drop. So from
Oh,
between day 5 to 7, this window,
yeah,
when we do the in vivo experiments and when we look at the echocardiography data, so basically to measure how good the heart is functioning,
yeah.
there is a very strong decline.
So, okay, so what is happening? Like what is that one thing? Like, what's the mechanism? I'm just like now I'm intrigued. Right? So there
Welcome
must
to
be
my
something
world.
that is happening, right? There must be something within these, like interactions that is happening, right? Or maybe something that is getting more mature so that they are not accepting these newer cells. Like, just wanted to know, like, is that the kind of thing that is
You're
happening?
thinking in the right way. This I can
Okay.
tell you. I mean, I don't have the final answer. There are
Yeah.
different schools of thought in the field right now. So
Oh.
some labs know that
Hmm.
in general, apart from the telomerase, what also happens in cardiomyocyte development is the metabolic shift.
Hmm.
So the cells that are that are contracting, they are quite unique in a way that they don't only depend on glucose for their metabolism. They
Hmm.
can also use fatty acids. They can also use purines and pyrimidines.
Hmm.
And the adult heart, so for us, like in humans, most of our cardiomyocytes depend on beta acid fatty acid oxidation. So
Hmm.
this is another way in which the mitochondria in our cells is able to produce ATP.
Hmm.
And this is very true for cardiomyocytes because they need a lot of energy for having this constant contraction and relaxation cycles.
Hmm.
So it's a very energy rich organ, our heart.
Hmm.
And therefore, to make it more the efficient, to make the energy metabolism more efficient, the
Hmm.
mitochondria in the cardiomyocytes have this switch.
Hmm.
So when we do this differentiation in the cell culture and also in vivo experiments, people have seen that
Hmm.
from a young heart, mice heart, to an adult one, the heart itself is switching from glucose metabolism towards beta acid or fatty acid oxidation,
Wow.
which is a metabolic switch that happens.
Yeah.
So less glycolysis, so more glycolysis when they're young, and
Yeah.
less glycolysis when they're old, and more fatty acid. This also correlates exactly to the time when this telomerase switch is happening.
Oh,
So
wow.
we
Hmm.
think that these two are like sort of connected to each other in
Hmm.
a we don't know how at this moment, but at least they
Yeah.
happen in the same time window, which then pushes the cardiomyocytes to become more adult in terms
Yeah.
of their physiology.
And this is how we are also, right? Like once we have a habit, we don't want to leave that habit, right? I mean, humans also behave that way, right?
Yeah, for some
You
it's
need to
good,
do
for some it's bad.
something,
Yeah, yeah.
right? And
And
I'm
the
assuming that this change in metabolic shift, it's also irreversible.
Not really. So in
Oh.
many of the heart failure cases, people
Hmm.
have seen
Hmm.
that when the cardiomyocytes are not healthy enough, they go back more towards glycolysis. But what does not change is another point is the polyploidy. So this is also very well known, even in humans, and it happens in all the mammal
Hmm.
kingdom. So mice, rats, whichever, hamsters, monkey, whichever model you will take.
Hmm.
The young cardiomyocytes are mononucleated, so they have only a single nuclei, which is also what we study in textbook, that you have one cell and one nuclei.
Hmm.
But the more adult or mature the cardiomyocyte gets, they become bi or polynucleated. That means one cell has two or three or four nuclei in this. This is polyploidy.
Hmm.
This is also a very specific character of mature cardiomyocytes.
Hmm.
What we have observed in our studies is once they become this polyploid, because we can also see that happening during our stem cell differentiation in culture. So
Hmm.
if I have the cells around day 20 or day 30 of the differentiation, 90% are single nucleated, so mononucleated. But the longer I keep them in culture, they do become polyploid. And
Hmm.
whenever I do these telomerase reactivation experiment, or whenever I try any other mitogenic drug to force them to proliferate, the ones that are binucleated, they
Hmm.
don't divide. So the ones that are mononucleated, they do. They
Hmm.
are, let's say, you can persuade them, but the
Hmm.
ones that have already become polynucleated, binucleated, they don't go
Wow.
backwards.
So I'm assuming like there is something. So like when again, like this is all naive stuff that I'm going to speak. Okay. So I'm assuming that there is some sort of an activation energy that is happening in the mononucleated, but when you have polynucleated, that activation is not happening, right? Because maybe there are two nuclei and maybe they want like they want equal attention. I don't know, like maybe something like that.
Yeah, I mean, this, I think the cell division will get more complicated
Yeah,
because
yeah,
if you have any cell that is polynucleated, binucleated, that means they are going until S phase. So if
yeah.
you imagine the cell cycle, you have G1, S, G2, and M phase, and
Hmm.
S phase is the step where the DNA replication happens. So
Hmm.
if you have a polyploid or a binucleated cell, that means it could go to S phase, and then
Wow.
it stopped at S phase. It did not go into the M phase. And this is also something you clearly see if you just count. This is like a,
Hmm.
how do you say, criticism in a lot of these cardiac regeneration papers that people just use Ki67 as a marker to show that cell is proliferating.
Hmm.
But it's not the best marker because Ki67 will also stain any cell which is in S phase or G1 phase
Hmm.
because
Hmm.
it's an interphase marker. It's not a proliferation marker in that respect. If you really want to prove that a cardiomyocyte is dividing, you have to use PH3 or Aurora B, like which are really at the late stages of mitosis, to make the claim that yes, I really have the cell that is going to divide.
Wow. Well,
So
I mean, this is a complex thing. Very
I'm
complex.
sorry, I'm just
I
going
am
into
having
like
a
too
I am
many
having a
complex
heart
things here,
attack,
but the
guys.
questions.
But
Just by hearing this.
so these are some of the like So whatever she said, this is like 100 years of research thing.
Yeah. I mean, and this is so
Many, many
pivotal,
years of
right?
So
research,
it's
yeah.
very vast to understand,
It is so pivotal.
and
Yeah.
these are like very high quality research, and that we
Yeah.
do. And I have mostly I've seen in Europe and US,
Hmm.
people do this kind of research. So that gets into segue to one question: that
Hmm.
are we doing this kind of research in India? First, if
Hmm.
yes, where? If
Hmm.
not, why not? Like, what's the hurdle? Because we are the top most population dealing with cardiovascular disease.
Yeah.
Do
Um,
you think?
to be honest, I mean, I know like in InSTEM, so in InSTEM Bangalore, there are labs that are working on cardiac development.
Hmm.
I know in the Central Drug Research Institute in Lucknow, so Professor Shashi Kumar Gupta, he was actually the first PhD student from our lab here in Hanover Medical School. He has his lab over there now, so he's of course doing cardiovascular research. There are one or two labs in RCGB Trivandrum. I'm sorry, I don't remember the names of head right now, but there
So
also they are doing
regional
research.
center of biotechnology, yeah.
Hmm.
Yeah,
Regional center of biotechnology. They are in Faridabad also. Yeah.
Hmm.
so yeah. But I just know these handful of 4 or 5 labs that do it in the whole of India. In India, it's much more common to find and come across labs that work on cancer.
Hmm.
For obvious reasons, there are many, many labs which work on all different types of cancer.
Hmm.
But even though cardiovascular disease and heart failure patients are there, I
Hmm.
mean, I think all three of us know at least 5 people in our close group where
Yeah.
either the person or their parents
Family,
or someone in
Hmm.
the family
family, family. Yeah.
has heart failure problems.
Yeah.
So it's very hard to come across labs, research labs in India that are working on it. I know there are a lot of clinical trials that go on in India,
Yeah.
Hmm.
but labs that are actually actively working towards figuring it
Yeah.
out, not so many.
Yeah.
And it's
That
I mean, it's very interesting. I mean, despite it having such a potential to
Mhm.
actually, find some sort of a medicine, right, which can be patented and can be, it is actually, the theme of of India as a startup country these days, right? This is
because
like a very important problem,
so
right?
all these research, especially at least on the diabetes and heart failure, these are like heart conditions. These are like the top two. These more
Yeah.
because in almost 70% of the population.
Hmm.
Human research is like very common in India. You'll see lots of clinical research, lots of
Hmm.
patient samples and those kind of. But the facility to do lower models, like using lower vertebrates like mouse or rabbits or pigs.
Hmm.
So that thing is not that much like the way it
Hmm.
have been. That's not much. And I read like every day I read papers on heart failure. That's how I got to know her about her. I didn't know that she was doing heart research,
Hmm. Hmm.
but because I was looking for different 100 types of papers, and I got to know that I rarely get any papers from India, at
Hmm.
least the actual model systems of heart, rather than in human heart research, because there are people who do work on humans. I
Hmm.
know,
Hmm.
yeah,
Hmm.
I know some of the labs that do that, and that's really, I don't think that saddens me. It's just it's weird that why it's not happening, you
Yeah.
know?
I personally know of one reason which I can imagine is because if you want to do these kind of cell culture experiments, as I said, that the cardioma cells don't proliferate so much, these are expensive experiments to do. So if as an institute you want to hire a researcher or a PI who has enough experience, you also need to be willing to fund the lab a lot
Yeah.
because you would have to always produce cells because the cells you produce once they will not keep proliferating. Cancer cells, you buy one vial,
Hmm.
Yeah,
it can be shared with the whole institute. You can also share the cancer cells with the neighboring institute and keep working on it.
Yeah.
This unfortunately doesn't work out with cardiomyocytes because you have to produce them every single time or you have to buy them every single time from a vendor,
Hmm.
which makes the whole experimental procedure very, very expensive.
Hmm.
If you want to do it with mice and rat studies like models and in vivo models, it's more feasible, I would say, in that way.
I-
But
yeah.
of course, then you need a proper animal facility in the institute. So, like, not every institute has that in India.
Hmm.
So, only big places like TIFR or NCBS, INSTEM, CDRI, IGIB, IITs, if they have an animal facility, would be able to do it.
Hmm.
Yeah.
So, I think this is also one very big reason why we still don't have
But
so many labs in India.
about these topics, my always think about is it's the mindset first that okay, so for next 10 years we should think about this. And
We
then
should. I mean, India right now is a young population, but it's going to be old very soon, right? And we will have these kind of. I mean, it's irreversible. I mean, that's a learning for me today. Everything in cardio is irreversible. So when it is irreversible, it's like might as well start working on it, right?
yeah, one more
This
thing.
is why the only option, sorry,
No, no,
Yeah,
Avinash, go ahead.
please go ahead. Go ahead.
yeah.
I was still to say the only way you can reverse is to get a heart transplant, which is again not available for everyone, and it doesn't work for every patient.
Yeah, yeah.
So we need it. We need some other strategies to help the patients, and we have a lot of cardiovascular patients.
Talking about that, I also want to talk about one more paper that you've just published recently. And I think this is about where you examine telomere dynamics during COVID lockdowns, right?
Yeah,
In
brain
healthy,
plasticity. Yeah.
yes, healthy older adults. Please tell us a little about that because I think that's also really interesting research.
This was actually for us also very interesting because we did not expect to see anything.
Okay.
Because it was such a short time window. Because
Mm-hmm.
this was in collaboration with a Swiss partner, so Swiss team,
Mm-hmm.
where no one knew that COVID would come. So there was a study that was gonna take place
Mm-hmm.
where they wanted to see for elderly people who are in like old age homes
Mm-hmm.
if they are given music lessons, if they are getting better in terms of cognitive efficiency. That
Mm-hmm.
was the whole idea. That was like a whole different brain plasticity study.
Mm-hmm.
They had already started collecting samples from the elderly volunteers who were participating in the study, and
Mm-hmm.
then came COVID.
Mm-hmm.
So, still, the people who were registered for the study, they volunteered to give their blood sample.
Mm-hmm.
Then there was the lockdown phase, and then we had samples again from them.
Okay.
So, the study was not designed for looking at telomere length, but it so happened that this coincided with COVID-19.
Mm-hmm.
And because we got the samples, we were like, okay, let's see what happens to the telomere length. Does that change or not?
Oh, wow.
And for us, it was super, super interesting to see that during the lockdown phase, there was such a huge decline in telomere length in these elderly patients, which then again, after like the restrictions were gone and then people started to meet each other,
Mm-hmm.
it also then helped to come back up. The striking thing was that it happened in such a short time. Because
Mm-hmm.
normally, as I said, over the year, you will gradually lose your telomere length. It should not happen like in 3 months that you lose such a huge amount.
Mm-hmm.
That was very interesting to see how psychological stress can
Mm-hmm.
also have such a huge impact on telomere lengths that you measure from the blood because we did not look at brain cells. We were just looking at blood samples and what is the telomere length in them.
Wow.
And
Mm.
first, we thought maybe we had a technical error in our experiments. Samples got mixed up or something went wrong because, of course, samples were taken from the donors and volunteers on different days. Everything was not done on one day. So
Mm-hmm.
we back calculated and we made corrections and correlations with the time of sample collection, with the days, with the months. No matter how we controlled for it, it always gave us the same result that during lockdown, there was a rapid decline in telomere length, which
Mm.
then got normalized afterwards.
Wow.
So
So
this
psychological
also tells
factors. Yeah.
that it had, at least for the elderlies, it had a huge impact. I mean,
Mm.
I cannot say this would be as drastic for young people
Yeah.
because, of course, young people also have,
But have you guys
other
done
ways to
any
connect to people.
research with young people though?
So, not really only looking at young people. We always get these, let's say, young samples from young people who are healthy volunteers to
Mm-hmm.
have age match controls.
Mm-hmm.
But we never had a study where we only looked at young people.
Yeah. Because I think
Because for
what
this,
would
we
be
again need ethical approval, and then we need to
absolutely,
have a
and
very
we
specific watch.
don't want another COVID-like situation. So I'm glad you're not going to do another study around that. But I think it will be really interesting to see like if there is a drastic dip with young
Yeah.
people as well, and is there like a pattern that's being because again like a lot of young people who are working these days they are also under a lot of stress. So I'm now worried about their telomere length as well, right? I'm worried about everyone's telomere length today, guys.
Okay.
I think,
So,
yeah.
we were talking about telomere and how some of the things are irreversible, and one more thing is irreversible is time. So,
Mm.
we've taken lots of our time. So,
Yes,
just
yes,
tell
yes.
me any new thing you are working on, or we should look forward.
I think One new thing we definitely work a lot in our lab is to, as we discussed now, that we don't have good models. And if you look at only the mouse or in vivo data, a
Hmm.
lot of drugs that are actually screened in these models, they don't work in the humans afterwards in the clinical trials. This is
Yeah.
well known for cancer, for any drug that you take, any disease you take, this is a problem.
Hmm.
So in our lab, a huge focus right now is to have human translatable models, one
Hmm.
of which is based on stem cells, of course. But then, if you now discuss models for cardiovascular drug development, cardiomyocyte is not the only cell in the heart. You have also the fibroblast, you have also the endothelial cells, which make the blood vessels. You also have neuronal cells that come into play. You have a lot of immune cells that are important, very important for the heart function.
Hmm.
So our lab is really focused on also building three-dimensional models, so
Hmm.
cardiac organoid models, or using uh, what we call as ex vivo living myocardial slices.
Hmm.
So when there is a surgery happening in the clinic, let's say there's an LVAR transplantation happening, during the surgery, the surgeon has to take out a piece of the heart to fit in the LVAD for the implant. And then this small piece of heart we can get as for research, and then we can sort of slice them into very thin sections. We
Hmm.
call them living myocardial slices. This is why,
Hmm.
and then keep them in culture and test the drugs directly on that.
Hmm.
So, of course, in this kind of scenario, you don't have a human healthy control, but you already have a diseased heart, which you can put in your
Hmm.
incubator in the lab and test the drug that you know is in the market. Or you can, if you have a new drug coming up, you can test if the drug is going to be toxic or not. So, these kind of things can be done directly on the human heart. And
Hmm.
in the stem cell models, we are developing these organoid platforms where we don't only look at the cardiomyocyte, but we also have the fibroblast, the endothelial cells, the immune cells all together. And then we look, if we put a drug, now what happens not only to the cardiomyocyte, but also to the other cell types of the heart, which will be affected, because when you take a drug as a human being, it goes everywhere.
Yep.
So
Yep. Yep.
this is one thing we really focus on in our lab a lot. And other than that, I think personally, the gut microbiome axis has a huge role to play when you think
Yeah.
about drugs and even health in general.
Yeah.
So
Yeah.
that is something people have not really looked into so much in terms of heart failure, in my opinion. And
Hmm.
I think there should be more research on how the gut axis also affects the heart
Yeah.
function.
I think just listening to you, I realized that there is one more experiment that can be done. I don't know, maybe it has already been done, but I think so. We have a lot of, people who are going to space station these days, right? So I would love to see if there is an impact on telomere length when
Been
there
done.
is being done. Oh wow, 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 wow. But is there an impact though?
It's actually in the US, so I think it's from Stanford. They have the
Uh-huh.
space. Getting the name again.
Yeah,
There is a whole space agency department
yeah,
there,
yeah, yeah,
and they are not only sending human stem cells into the
yeah,
space
yeah.
and then looking at telomere length,
Hmm.
but they also did with the astronauts. So
Yeah,
this is known that if you
yeah.
go to the space and you come back, you lose telomere length. So they
You
published
do.
it in 2024, end of 2024, beginning of 2025.
Wow.
I can share the papers, but
Yes,
this data
please. I
already
think that
exists
would be
and they did it
wonderful
with the
because
astronauts.
it's really interesting because, like, as human beings, I think the next frontier that we want to achieve is to get in onto Mars or maybe, have a space station on Moon. Because, and if we're going to do that, we need to know how,
Are not
our-
good for the telomere lens. This I can tell you.
fair point. Let's not tell
Yeah.
the
Let's live
astronauts
here.
this.
Eat healthy and let's live here.
Okay.
So first of all, thank you so much. This was very insightful, and in 40
Yeah.
minutes, you have covered a lot.
I'm sorry, it was too dense with a lot of information.
So certainly not
But
for
these are
me.
like topics I am so involved in, so it's
Yeah.
hard for me to control
No,
myself.
no, we'll
No,
we'll
no,
talk
but this is
again.
what we wanted, right? I mean, this is the reason why we are, building this as well. We want young scientists to come and talk about their work, right? As deeply as possible, because otherwise, at the end of the day, we are only looking at scientists who win an award, right? Or who've like, at the helm of everything. But why not, talk to young scientists like yourself and talk about what is it that's actually been done.
So,
Yeah.
so
Hmm.
any so what we do in our podcast at the end all the time, we tell to give some message about science or scientist in the native language, and you speak Bengali. You
Yeah.
speak Hindi, you speak German and English. So I don't know
I,
what your native language is. So I don't know.
My mother tongue is Bengali.
hmm,
But
So,
yeah, I mean, as I said, I grew up in Patna and Jharkhand, so Ranchi, so I'm as comfortable in Hindi as well.
so
So
whatever you are comfortable in, give a message, a one or two line, and then at the end, just tell how was your experience here.
yeah.
I guess I will do it in Hindi because then there are more people who can follow it, if that's
No,
fine.
like it's fine. We have done in Marathi, Bengali, everything. It's up to you. It's up to you. Your choice.
I mean, I can do it.
Yeah,
Yeah, I mean, in Hindi, if following up on all our discussions now,
yeah,
and as Mrityunjay kept saying, like, oh, this is that he's scared now for people's telomere length. My personal feeling is that whenever I talk to people, and especially people who know that they're not living a healthy lifestyle, I keep getting this answer like, क्या होगा अब बदल के इतने दिन से तो ऐसे ही चल रहा है अब
Hmm,
बहुत देर हो गई है कुछ बदलने के लिए सो मेरा ये मानना है कि इस धारणा को चुनौती देनी चाहिए
hmm,
शुरुआत करने के लिए कभी भी देर नहीं होती जिस
hmm,
दिन सोचो जब जागो तब सवेरा तो अगर
hmm,
आज महसूस हुआ है
hmm,
एहसास हुआ है कि नहीं अभी तक कुछ गलत था या अच्छा नहीं था
hmm,
तो उसको सुधारने की कोशिश करनी चाहिए
hmm,
मेरा यही कहना है तो अगर आपका हेल्थ है या आपका पर्सनल लाइफ है या प्रोफेशनल लाइफ है जो भी है
hmm,
कभी भी ये नहीं सोचना चाहिए कि बहुत देर हो गई है जो
hmm.
हुआ है उसको आप बदल नहीं सकते पर आगे जो होगा उसको आप जरूर अच्छा कर सकते हैं तो मेरा यही संदेश है
yeah. Thank you for personally attacking me.
I mean, I am equally responsible. As you said, I hardly sleep well, so guilty as charged.
I know, I know. So that was this was incredible, and I hope you enjoyed our conversation.
Yeah, it was a wonderful experience.
Yeah.
I'm sorry I also went overhead over the
No,
time, but
no, that's totally all right. I think I think we are taking a
I'm really connected to this topic, as I guess you have
so
understood by
I
now.
think like as someone who's not a biologist, as someone who doesn't understand a lot of biology, I think that this it's really fascinating, right? And that's why like even like someone as I said, I don't understand biology at all. But I had a lot of these questions because I feel this is something that's related to our daily life, right? And very important for everyone. It doesn't matter, like not just India, but everyone across the world is facing the same kind of problem, same kind of stresses, right? So I mean, we are thankful to you, coming in and talking to us about this in such a in such details, right? So thank you, actually.
yeah. So thank
I'm
you so
happy that you could follow it. I mean, as I said, I went too much into details, but
Yeah.
but your questions were all really valid. And as we discussed, like there are questions that either people are researching or they have already researched. So
Mhm.
all valid questions.
Yeah. So those who will listen to it till the end, like, share, subscribe, and also learn more about science. Talk to
Mhm.
people about science. Talk about health and how to live better, and have curiosity about science because that's what this platform is. And this is about positives about science, about people who has done good and who are doing good. And I'm waiting just a professor in front of Shambhavi Chatterjee as soon as possible because she already has credential more than many people. I know I'm in the field of art, so I can say that. So I'm waiting for that. Fingers crossed for that. Anywhere in the world, because science is not restricted to a place. And just like, share, and we'll meet you in the next episode.
Yeah.